start recording again. Okay, so we're really pleased today to welcome Dr. Struan Grant to our Knowledge Portal Network webinar series. Struan did his undergraduate and graduate work at the University of Aberdeen. Then he went on to do a postdoc at the Garvan Institute in Sydney. He then worked at Decode Genetics in Reykjavik, and he's now the director of the Center for Spatial and Functional Genomics at the Children's Hospital of Philadelphia and the University of Pennsylvania. He's been working on human genetics and genomics for his entire career, and he currently focuses on the genetics of pediatric conditions such as childhood obesity, early onset diabetes, and more. We on the Knowledge Portal team have had the pleasure of getting to know Struan as our colleague in the AMP-CMD consortium for the past few years. We're looking forward to hearing his talk today on variant to gene mapping, which is, of course, one of the major goals of our project. Take it away.
Thanks very much. Thanks very much, and thanks for the opportunity to present, and great to see people on the line, some names that I recognize as well. So what I'm going to talk about today is variant to gene mapping for common complex traits, but in fact, many of our approaches that we're employing could actually also be applied to rare disease and rare variants as well, but really the focus is principally on non-coding variation, as I'll get into. I'll just click on my active window here. So my overview of what I'm going to discuss is GWAS, which is of course been around for some time now, and has really been bearing fruit, and there's been a lot of positive outcomes. But there are some shortcomings to the approach, which I'll also cover. But despite those shortcomings, the question I've been asking, and many of us in the field have been asking, is how can we harness what's been found to sort of advance understanding of the disease and potential therapeutics and diagnostics. And some of the examples I want to use to sort of show how we are addressing this is through sleep, obesity, bone, diabetes, lupus, and COVID. And really the way we're tackling this is through the sort of epigenetic route. I'm not claiming to be an epigeneticist, but some of the tools that epigenetics give us can be leveraged for the way we're going about doing this. And this is sort of how I'm seeing the progress in common complex trait genetics over the last, say, 20 years, or maybe a bit longer than that. But in the first instance, there was really candidate gene studies. And that was really the domain of molecular biologists. I myself did a PhD on a candidate gene with Stuart Ralston in Aberdeen, where we said, let's take a collagen, one of the collagen genes. Collagen is the main component of bone. Let's look for genetic variants in the lab, see if they're associated with osteoporosis. And so that was just focusing on one gene, and it was really the domain of the wet lab scientist. But then genome-wide approaches came in, where you could do a more uniform approach and sort of look across the genome in a non-hypothesis manner. First up really was linkage, where you looked at families and looked for regions of sharing across the genome within families. The problem with that was you couldn't really nail the causal gene. You still had a very large amount of real estate where the possible causal gene resided. GWAS really changed that, in that we had much larger case control sets. And that was really the domain of the clinicians, because we as scientists were very dependent on the clinicians giving us the samples. And then, then sequencing those DNA collections, we're really at the mercy, if you will, of bioinformaticians to be able to do their thing and analyze these large scale data sets. But really the reason we're generating all these data sets, these genome-wide data sets and billions of dollars worth of data in the public domain, is really to distill it down and to then go back to the wet lab and to commit to certain targets that are really attractive, and there's a huge body and confluence of evidence for these loci to really commit to understanding what these particular genes are doing. So I think, again, the molecular biologist has the opportunity to sort of lead the way in this area. In my own particular interest, as Maria said, is pediatrics, and I'm based at Children's Hospital Philadelphia and on the faculty of Penn. And really the way I've been attacking this since I came here was looking at pediatric routes to type 2 diabetes. So we know that obese children are more likely to be obese adults as opposed to the other way around, and therefore an increased likelihood of type 2 diabetes. Birth weight, if you're born small or large, you're more likely to have disease in later life, like type 2 diabetes. We're seeing an increased prevalence of type 2 diabetes being diagnosed in children. And there are other types of diabetes like type 1 diabetes, LADA, MODY, and these also present opportunities to understand type 2 diabetes. So that's sort of been the niche I've built for myself sort of academically. But orthogonal to that, I've also got more interested in sleep and how sleep can affect obesity and diabetes risk and vice versa. And as Mendelian randomization studies show, the effect is sort of two-way. And a bit more orthogonal to that is also, I'm also very interested in bone. I did my PhD on bone, got distracted by diabetes, but I still remain very, very interested in bone. And of course, there is a relationship between all these traits and an effect on bone and biology as well. So I'll be touching on all these as we go through the talk. So just to give you a vignette about my experiences with GWAS, again, in the pediatric realm, one consortium that was set up a few years ago was called the Early Growth Genetics Consortium, or EGG. And that's been an extremely useful collaboration where we've been able to compile all our pediatric data sets and find new signals in a meta-analysis setting. What was particularly interesting about this GWAS was that this found loci that were familiar to us GWAS geeks already, in that they already had come up in adult diseases. These were type 2 diabetes loci. This one's an adult blood pressure locus. These two are adult height loci, but they came up in birth weight. And so the idea being that perhaps these adult traits are actually secondary traits to the actual primary trait, which is much earlier in life, i.e. birth weight in this case. And in fact, as the birth weight GWASs have got larger and larger, this was the earliest GWAS. Since they've got larger and larger, this seems to be holding true that many of these adult loci are operating very early in life or maybe involved in development. Likewise with childhood obesity, I was lucky enough to land the lead on the EGG effort on childhood obesity. We continue to do that today, but this was the first iteration of the GWAS. And we found a number of loci that are shown in black here that were already discovered in adults. The difference was we needed a sample size, 1/10 of the size that you needed in adults to find them. So it was clear that these were more marked in children. They were easier to pan out the gold in kids. We also found 2 additional loci in red, which did ultimately come up in adults, but it was more readily detectable in the pediatric setting. But really what I want to bring your attention to is the FTO locus. So the top locus here, the Empire State Building signal, if you will, of this Manhattan plot. And what we've learned from FTO has been very important to my group and to many others. Really seminal studies have come out on this FTO locus, and how approaches were applied there really can be applied to many different traits. So definitely very inspired by those FTO studies. And initially, when the first GWAS came out of FTO, this German group very rapidly knocked these out in mouse and got this in nature. And what was interesting was that the FTO knockout mouse, which you can see on the right here, was slightly smaller and leaner, despite being hyperphagic and not moving in the cage. So clearly there was excitement in that this mouse had the lifestyle and the looks, and it presented itself as an exciting prospect for therapeutic intervention. However, things got a bit more complicated, as I'll come to here. Here's a shot of the UCSC browser, and you'll see these green dots here in the FTO gene, which really represent independent studies that have all seen FTO like we have in the kids, that there's very strong consensus there's this region in the intron of FTO. And so there's been a big presumption that FTO is the causal gene, and lots of studies have gone back and forth characterizing the function of FTO. However, these 3 studies that are shown in the corner here, coming from Chicago, MIT, and Columbia, really added context to this locus. So in the case of the first 2 papers looking in fat or adipose, there was evidence that, in fact, this region, this non-coding region, was actually driving the expression of IRX3. Furthermore, the MIT study not only showed IRX3, but also implicated IRX5. The Columbia study, which looked in the brain context, pointed to the gene on the left. And in fact, I think there's more and more evidence that this is quite temporal. Depending on the state of the cell, you can see different control of different genes in the neighborhood. So the pessimist would say, gee, this is more complicated than I thought. The optimist would say, well, in fact, for the price of one signal, I'm getting multiple genes. But it's clear that we need to go through each of our loci that we found in GWAS and actually understand what's going on at each of these loci and which targets are most interesting. And so really, to summarize what I've just said there is you've got a variant. It's typically non-coding when it comes out of GWAS. It's either in an intron or it's between genes. Is it driving the nearest gene or is it driving a gene further afield? Now, of course, there's GTEx, there's EQTL studies that have been done, and those are absolutely massive resources. But for instance, with FTO, that had to be resolved using much more hands-on approaches in the lab. And so EQTLs don't solve every locus. Plus, GTEx is made up of it's made up of tissue, so it's mixed cell populations. So there's still room for other orthogonal variant gene mapping approaches, but certainly integrating the two, which I'll talk about later, the approaches to try and get up a confluence of evidence is very important. Something that's become very interesting to me is TADs, or topologically associating domains. And this was described just over a decade ago by Bing Ren at UCSD. And this idea of regions of interactions in the genome are relatively local. So you have blocks of interaction followed by a boundary element and then another block of interaction and relatively little crosstalk between the blocks. That's put very simply. And if I show you a cartoon of the next, of how I'm sort of thinking about TADs, it's really talking about the search space. How far left and right of a GWAS signal should I be looking at to find the possible causal gene or genes at a given locus? And so in this cartoon example, if we look at the top panel, the red gene in this case is the causal gene, but the blue gene or the blue region is the region that we are pursuing. So in the case of the nearest gene, which has been very typical up to now in terms of nomenclature, that would be the wrong gene. If we looked at an arbitrary window and said, well, I'm only going to look 100 kb either side of the signal, again, I'd have this blue window, but I'd be missing the gene. Whereas if I look at the region of actual interactions or the TAD, then I would capture the red gene. I'd get the causal gene. And what's really particularly attractive about TADs is that TAD boundaries seem to be principally tissue independent, i.e. the boundaries are the same irrespective of the cell type you look at. So it's been helpful, but not crucial for me, but it's been helpful, and our approaches do obey TAD structure in that our search space is pretty much, in terms of looking at an effect on cis, housed within the TAD. So the first example I want to give you and how it really got me into this follow-up game as opposed to just being a GWASer was in the context of type 2 diabetes. I was working in type 2 diabetes many years ago. I'm not so active now on adult type 2 diabetes, but I did have an early success working in type 2 diabetes. This is one of the more recent GWASs of type 2 diabetes, and the sort of Empire State Building signal, the top signal for type 2 diabetes is at the TCF7L2 locus. And what was a really good day in my career was whilst I was working at Decode Genetics in Iceland, which we published from the company. We first reported the association with TCF7L2 and type 2 diabetes. So it was very satisfying to see when the first GWASs came out, because we used this using a slightly different approach, but when the first GWASs came out, it was very satisfying to see TCF7L2 as a top signal. And as you can probably imagine, I'm very invested in understanding more about this gene and getting funding and pursuing this particular locus. And so this was sort of the first adventure I had with my lab to sort of explore it. And this is the TAD region for TCF7L2. And in fact, it's quite a gene-rich TAD. There's a lot going on in this region. And so if TCF7L2 is not the causal gene, is it still worth pursuing? And I'd say the answer is yes, because there's a number of genes in there that are therapeutically potentially attractive. And so trying to understand exactly what the landscape is with respect to operating genes is very important. And so what was what had caught my attention was that there was very strong consensus in the field generally that the causal variant for this, the underlying association, had already been found, and it's this SNP here. Again, sitting in an intron, a bit like FTO, sitting in an intron of TCF7L2. So given that, the field had consensus on this potential causal variant, we decided to make our first wet lab adventures. And so what we decided to do was not as elegant as the MIT study for FTO. We took a colorectal cancer cell line called HCT116, which is a obviously a pretty messed up cell line. But TCF7L2 is a known colorectal cancer gene. That was part of my logic. The other part of the logic was they're very easy to CRISPR. They're very easy. It's a very easy cell line to manipulate. So this is really just our first attempt. And what we did was we did CRISPR vandalism. We basically threw a few CRISPR grenades in around the region of the SNP and then asked, what does it do to the gene expression in the neighborhood? And so we had a 66 base pair deletion, 104 base pair deletion, and a 1.4 kb deletion just around that SNP. And this is what we saw. So we saw on the right, this graph, TCF7L2 expression was affected. So it was down, but it wasn't as marked as we saw with a gene 3 genes away, ACSL5. And in fact, this is RNA at the top, and at the bottom is protein. This is the Western blot. And as you can see, the ASOL5 expression is almost fully ablated, irrespective of the size of the deletion. So that was very interesting. So what we concluded was that this SNP is sitting in the intron of TCF7L2 and is influencing TCF7L2. But we're also seeing an effect on the expression of ASOL5. Now, there's some caveats to this, and I think this is our experience with this is typical of what people are experiencing and how you address it. And it also poses more questions than it answers. First, I used a fairly crappy cell line. So what's this doing in other cellular settings? EQTL studies of this region in pancreatic islets, which is obviously a different cellular context of what I studied, only show TCF7L2. So it's only an EQTL for TCF7L2. The other thing is TCF7L2 is a transcription factor. So it's conceivable that, in fact, by messing with TCF7L2, it has a trans effect on ASOL5. And that still has to be fully elucidated. That is this purely cis, or is it purely trans, or is it a combination of the two? So there's still quite a bit to work out. But certainly, what's exciting about ASOL5, it's an acyl-CoA synthetase, so it's actually a druggable target because it's an enzyme. And already before we even did this study, a knockout existed at Tufts, and the mouse is indeed diabetic. So this spurred us on to go to the Children's Hospital of Philadelphia and said we wanted to start an operation centered around spatial and functional genomics. At that point, we were thinking of spatial from a 3D genomics point of view, genome folding, but spatial is now sort of really more the domain of single cell. So I don't want to confuse things here. This is not so much single cell as it is to do with 3D structure. And so our variant gene mapping at scale is very much influenced by that FTO study, where on the panel on the left is looking at linkage to this equilibrium. So trying to be as liberal as possible in our selection of genetic variants. So taking the top variant at the top of the Empire State Building signal and all the other signals, taking all the SNPs and linkage to this equilibrium, and then saying one of this series of SNPs at this given locus could be causal. We then impose a tag-seq on that by just saying which of those SNPs in that proxy series are open. Typically, the top SNP is not the causal SNP, and it's typically not open, but some of the other proxies are. And then we say, of that subset that are open, which of those are contacted by a promoter? And it has to be done at high resolution, as I'll come back to. And then we're down to just 2 or 3, maybe 1 to 3 variants per locus. So we've got down to a manageable set of possible causal variants. We've made implications for a candidate gene or genes at a location. But we don't stop there. We really do like to go on and then do sort of functional analysis, at least for a subset, to verify that what we're seeing is it can be validated. And so that's where the cellular studies are done. And there are a number of chromatin conformation capture approaches out there. we were very taken with these papers, and they're all quite different. So the paper on the left, from Cell and Peter Fraser, they did high-C for immune traits and implied a number of different genes. The concern about that is the resolution. You know, that if you're using a low resolution high-C, you're looking at big chunks interacting with big chunks, so you may not have the resolution. So that was something we debated. We also debated high chip, which came from Howard Chang, but that's obviously very, very dependent on particular marks. And so there's a sort of a priori expectation that certain histone marks are crucial to this. Whereas what we liked about Capture-C, which was published by Oxford, was that you can really dictate which regions of the genome you want to find what they look for. And so we thought we'd try and scale up Capture-C for our own uses. And so what we decided to do was put a probe in every single promoter in the human genome, along with every non-coding gene. So microRNAs, antisense RNAs, links. And so rather than making probes for SNPs, because obviously more and more SNPs are being discovered for disease every day, rather we would go from the gene out. And so we could then, ipso facto, if a SNP came along, we could ask, is that promoter interacting with that SNP? That was the idea. So what we wanted to do was generate a map which was 36,500 RNA-baited fragments, and it was really a one-stop shop. So if we generated this in a cell type, we could go back and query it as more SNPs came along. And of course, it required very deep sequencing, and so we used the NovaSeq platform to do that. When I talked about resolution, this is pretty important for us, and Chun Su in our group studied this. And so looking at the promoter capture high-C I was talking about with that Cell paper, that was done with HIN3, which is a 6 cutter, so it's a relatively infrequent cutter, and we were using DPN2, which is a more frequent cutter, and so we just chopped the genome up in smaller chunks. So what we observed by doing a head-to-head comparison was at the bottom here, the HIN3, the bigger chunks, we could only see these sort of more distant interactions, whereas with the DPN2 red regions, we could see many more interactions nearer the bait, near where the probe was. And so we could get around that potential myopia that was in lower resolution high-C. And so if it was the nearest gene at a given locus, then we more likelihood of seeing it. So, the first disease that we applied this to was lupus. The reason for that was my close collaborator and fellow director of this operation, Andrew Wells, is an immunologist. And when we were talking about lupus, he said, you realize there's a plentiful supply of T follicular helper cells at CHOP because of tonsillectomies. And so TFHs are one of the primary candidate cell types where lupus variants could be operating. So by applying this approach, Chun Su again analyzed this data. One of the key loci for lupus, one of the top loci, is at the LPP gene. And a lot of excitement because LPP on the face of it looked pretty novel. But when we looked at the proxy SNP in LD with the GWAS SNP, we saw that in fact there was interaction with an open promoter or an open SNP in the LPP gene with an open promoter with BCL6 really on the other side of the TAD. And to Andrew, that was unexciting because BCL6 has been known for some time as being a key transcription factor in T follicular helper cells, and the biology is very well known. So in a sense, in this case, we kind of said, okay, well, we actually found known biology. Yes, it's a new regulator harboring a SNP some distance away, but the target gene is already known, so we can move on and look at other loci. And by doing this as a larger scale, Andrew is not a GWASer, and he finds it hilarious that us GWASers do name our loci after the nearest gene. And he said, well, what if I take those nearest genes to all those GWAS loci and do pathway analysis? Only 2 of the top 10 pathways are relevant to TFH biology, whereas taking our pathway analysis, or taking our analysis with what we think are the more closer to effector genes, at these loci, 10 for 10, were relevant to TFH biology. Now, of course, we carried this out in TFH cells, so that's one point, but these SNPs were not discovered in any particular tissue context. That were found in blood-derived DNA, presuming that they're in every cell type in the body. But it did feel like we were closer to the biology doing this with our approach. What I also pointed to was that there was a lot more known biology at these loci than what was originally sort of seen when you looked at the nearest gene. That meant that we could actually triage a lot of the leads away because they'd already been studied, and it left us with a handful of genes that had never been implicated in lupus. And we started working on those, particularly in Andrew's lab, and we actually realized there's a couple of genes where there's existing compounds that haven't been applied to lupus, and we're sort of exploring that route. So coming back to the diabetes route and these orthogonal traits that I'm interested in, bone mineral density has been, as I say, an interest of mine since doing my PhD in Scotland, and you know, I got back into it when I met Babette Zemmel at CHOP, who's been looking at or defining with the NIH pediatric growth curves for children, both with respect to BMI and BMD. And so we were lucky enough to get the DNA on their final visit and then to be able to carry out GWAS. And one of the loci that was very striking to us was the bone density at the wrist, a common region of fracture for kids. And so we found this top signal on the left here by Alexander Casey. And this signal had also been reported for, I think it was spine and hip in adults. So a well-known bone locus. And the signal resides over CPED1. And as you can see at the bottom of this locus zoom plot, and a lot of people in the field think, well, it's either CPED1 or it's probably the gene on the right, Wnt16, because Wnts are very much sort of been implicated in bone biology as been in endocrinology generally. But we thought we would leverage our approach, carry it out in HM's human mesenchymal stem cell derived osteoblasts, working with the University of Michigan and Kurt Hankinson, and see what we saw at this location, along with every other locus that's been reported. But staying with the CPED1 locus, the green bar you see on the right, ending with the SNP 9O, that's the top SNP, the sentinel SNP for us with the kids. It's not open and it's not looping to a gene promoter, whereas two SNPs in very close proximity to each other, actually they're so close to each other in the same attack-seq region, was open and was contacting two gene promoters. It wasn't Wnt16, it was CPED1 and ING3. And ING3 really hadn't been on anybody's radar with respect to this region. By then working with the University of Michigan, again, Kurt Hankinson and his excellent team member, Yadav Wagley, they could derive HMSC derived osteoblasts, knock down the expression of our candidate genes, and then see what the effect was on measures of osteoblast function. And this is what the data looked like with Kurt's help. We decided to take all three genes out, ING3, CPED1, which were implicated, and Wnt16, a primary candidate gene in the region. And in our hands, we found that ING3 knockdown had the most pronounced effect, that there was less blue, which means there was less alkaline phosphatase being generated, a marker of osteoblast function. And also there was less mineralization indicated by the red, alizarin red staining. Now, we're not saying it's not CPED1 and Wnt16, and we know these effects can be pretty temporal. Depending on the state of the cell, these effects can come and go. But certainly in the conditions that we were working in the state of the cell on that day, the way the sun was shining on those cells, we're ruling in ING3 as being one of the effector genes of this locus. Another complication that Cheryl, who I know is on the call, has pointed out to me that CPED1 is highly alternatively spliced. And it could be that we didn't address every single splice isoform, and that's why we missed it there. But the idea being that there's possibly, the reason this is one of the top loci for bone is because there's maybe quite a lot going on. But we are pursuing ING3 as being at least part of the story at this locus. And just as an aside, as I said, with Babette Zemmel, she'd actually been collecting these kids over years, and what helped us actually find more loci, than was the fact that we had this longitudinal information that we could take multiple time points, and by doing longitudinal, it helped increase our haul of genetic variants for bone mineral density in kids. The other trait that I referred to was sleep. And sleep's an interesting one because it's so highly conserved across organisms. And so Philip Gurman at Penn sort of introduced me to Alex Keene, who's now head of biology at Texas A&M, and is a fruit fly and cave fish expert. And so we engaged him on the fruit fly aspect and said, is there a way that we can take our predicted genes from our approach and test them in fly? And so the concept being is we take the human insomnia GWAS that was already published, I had nothing to do with. Let's take the candidate cell type, in this case, neuroprogenitor cells that were derived from iPS. We happen to have our chromatin promoter maps generated for that. And then say, let's generate a candidate gene list. Let's knock them out in flies. And those that look interesting in the flies, let's then subsequently knock them out in fish. This is what we saw. So we had 54 genes that had orthologs in fly. Some of those flies had multiple RNA pan-neural options. So we had a total of 73 RNAis. And then this is what we saw. So, or what Alex saw. So you can see these gene names in red. So at the right-hand side, we've got a number of genes, and these are long sleepers. These are very sleepy flies. And at the very left here, the other part of the extreme, we have very short sleeping flies, or I like to call them hypercaffeinated flies. They're very full of activity. And so it was interesting that we saw this from this distribution, we saw these genes exhibiting this behavior. One of the early hits that we got was PIGQ, and so we have a head start on PIGQ, and we've published this paper really focusing on PIGQ. And there was 2 RNAi lines for PIGQ. That's why I've got 2 panels here. I won't go through this in any great detail, but when we looked at different metrics of sleep, clearly was having an effect in the fruit fly. And then Amber Zimmerman in my group that I co-mentor with Alan Pack, we also saw an effect on zebrafish. And in fact, the zebrafish also became long sleepers. Something else that's become interesting and becoming more crucial is also that there's a whole family of pigs. I wasn't familiar with this gene class until this study. And in fact, when we perturb many of the pig genes, we see the same phenotype. So it's really led us to a new pathway that we're focusing on in the context of sleep. A key cell type for many of the brain traits and for metabolic traits is the hypothalamus. And so we thought it would be a very good move to generate our data in the hypothalamic setting. And Matt Paul and my group collaborated with Rudy Leibel up at Columbia, and we generated an embryonic stem cell derived hypothalamic neurons. So we looked at the ESC, the progenitor, and the full mature neuron, and we did our trifecta of RNA-seq, capture-seq, and attack-seq. And then we made causal gene implications. And so we implicated a set of genes. The interesting thing is, when we looked at our RNA-seq data from the cell model setting, the greatest correlation was actually with hypothalamus tissue in GTEx. So this was what our data looked like. In this case, we decided to do LD score regression, which is really taking a bunch of GWASs for different traits and asking, are those signals enriched in our features, i.e. open chromatin and being contacted, a CRE, as we call them. It's a contacted open region. And where you see stars in these circles was where there was a significant enrichment. And why we were doing this was, rather than guessing which traits we should study in the context of the hypothalamic data that we had, why not have the GWAS data tell us which ones are enriched? And so in this case, we saw enrichment for body mass index. Not really a surprise there. We know hypothalamus is very crucial to BMI regulation. We also saw age of menarche, very strongly enriched in all three settings, as we did for height, for chronotype, and other metrics of sleep. So these are traits that we would then be motivated to do variance gene mapping at the individual level and perhaps do functional follow-up, given the guidance that we get from doing this LD score regression. So we've had a proliferation of grants. We've had a good time working with the NIH on a number of different disease areas doing variance gene mapping. One is Alzheimer's, and we've been working with Jerry Schellenberg and Lisan Wang. Alexander has recently left my group to become a faculty member over with them in pathology at Penn. We're still working very closely together, and the idea being as they generate the bigger and bigger GWASs for Alzheimer's, we are in a position to immediately sort of take those signals and try and add context to those signals. Type 2 diabetes is another one we have funding for, and there's 6 PIs in this effort. There's myself, Patrick Seal, Wenli Yang, Ben Voigt, Klaus Kestner, and Dan Rader, and we're all doing various different approaches here. Ben, myself, and Andrew Wells are doing the sort of computational part, making those calls with respect to EQTLs and chromatin capture. And then we're passing them over to these highly talented functional biologists who are working in different systems like liver, adipose, pancreas, and then seeing what happens when you do a functional readout on those loci. We were not the only awardees. The other awardees was a sort of combination of people from Stanford, Harvard, UNC, Michigan, etc. And in fact, we're having a good time collaborating with them as well, where we're all working together and doing gene nominations in what to pursue. And given there's now many hundreds of loci for type 2 diabetes, there's an embarrassment of riches in terms of what to pursue. This was one of the first papers that came out of that effort. Again, Chun Su and my group did the analyses, and it was co-led between myself, Andrew Wells, and Klaus Kestner. We made the cover, and Ben Voigt's wife designed the cover. And we think it's a really cool cover, but it sort of also exemplifies what we were doing. We were taking the pancreatic islet cells and looking at them in a sorted way, so we could look at beta cells in isolation, acinar cells, and alpha cells in isolation, and then see what interactions we saw. And again, what I showed you with the hypothalamus, we could sort of say, well, where is an enrichment? In this case, the red means it's significant. So if we look at the acinar alpha and beta as outlined below, you can see where there's significance. So in the case of fasting glucose, we see fasting glucose GWAS, we see an enrichment in our features for all 3 cell types. Whereas type 2 diabetes, we see it only for alpha and beta, which kind of squares with what is being seen in the field generally. Hemoglobin A1C seems to be almost entirely beta cell, whereas pancreatic cancer seems to be entirely acinar. And that fits actually with what the field thinks is the acinar is the primary candidate cell type that confers the effect of the genetics. Whereas chronotype and sleep duration seem to be showing different things in different cell types. So we're interested in this, and I know I'm talking to the portal team here. This is data that we've generated as part of that grant, and that's now being deposited in the portal for all to query. But what's great is when we're generating these data, and we have this one-stop shop. As I said earlier, when somebody does a GWAS, we can potentially just sort of immediately query our data and add context. So this is a GWAS of NAFLD, which was principally led out of Penn. And in addition to the GWAS, they could query EQTLs and our liver data that we generated in the same way. And so then be able to make a query. And if I can just add one thing, actually, the type 2 diabetes paper I mentioned there, the pancreatic cells, that actually was done with Hi-C. The reason for that being is there's now new Hi-C methods and commercial solutions that have the resolution that we like. So we're actually rapidly moving from Capture-C to Hi-C, given we're happy with that new resolution. And another vignette of what we did was, again, we actually have a grant studying type 1 diabetes. We're working with Steve Rich at the University of Virginia, trying to add context to type 1 diabetes loci. But given we've created these maps in all these different immune cell types that are relevant to type 1 diabetes, we can also pivot to other immune traits in those same cell types. So let me show you an example. So this is COVID severity, a recent GWAS, obviously very topical. And we took those loci and asked, well, what does our data point to? I won't go into the variant gene mapping piece, but I will talk about the LD score regression again, just asking which of those loci are enriched in our features. And what we find in our hands is that the loci en masse are enriched in naive T cells. So that's what we see. So overall, we have in the field an arsenal of approaches. You know, I'm not the only person doing this. There's a bunch of people doing this. I know the Broad is making a big commitment on variant to function analyses, and there is a myriad of ways that you can do this. There's many algorithms now, like the ABC algorithm that's being used by many, there's EQTLs, and there is approaches like we're using, chromatin capture, CRISPR-mediated deletions, CRISPR screens, and then ATAC-seq, and then increasingly single-cell approaches. But I think ultimately, this is a paper I published with Casey Brown a few years ago and won an award at ASHG. And the reason I bring up that is, of course, Casey recently passed away, which was very tragic. But we had a very good collaboration doing this by integrating EQTLs, and him being a sort of key leader in the GTEx consortium. We found that we could generate a confluence of evidence for certain genes by integrating EQTL data with our chromatin capture data. And the last point I want to bring to your attention, and something I've been thinking about, and it's still a little half-baked, but Reza here in my group published this paper in eLife a couple of years ago. And the question was, can we take existing GWAS data sets and actually salvage loci that are actually not strictly genome-wide significant? Can we pull out things that are below 5 × 10⁻⁸ by putting these constraints on sub-threshold GWAS hits? So let me just orientate you for a second. So just imagine you've got a series of GWASs that have been published for body mass index. First one, I think, was 2000. Well, one of the main ones was published in 2010. There was another one in 2012, another one in 2015, and they got bigger and bigger. And the question was, well, if we go back to that 2010 GWAS, impose our features, and then ask which of the sub-threshold signals fulfill our criteria, did they subsequently become genome-wide significant in a larger, later GWAS data set? And in fact, it was quite successful for us, that we found that we could rescue signals that were published much earlier on and were subsequently genome-wide significant. So we think that there might be a trick here, maybe using our features and maybe other types of features. By imposing this on existing GWAS data sets, we can perhaps rescue sub-threshold signals and not actually have such a major need to collect larger and larger sample sizes. I know that's a big statement, but I do think there are tricks that we could employ as a field to try and rescue and try and find that missing heritability. And my very, very last point, I forgot about this slide, but my very, very last point, talking about missing heritability. This is a paper that came out in Nature in last year, led by Joel Hirschhorn at Boston Children's, and I'm proud to be a co-author on the paper. This is really the first GWAS where you've hit saturation, at least in European ancestry. The idea being it's such a large GWAS with 5.4 million individuals that, in fact, if you collected any more individuals, you wouldn't actually find anything new with respect to common variation. So effectively, the genetics of height in terms of common variance is solved. Now, what does that look like? It looks like there are over 12,000 loci, and if you consider the block of linkages to equilibrium on average around them, you're looking at about 21% of the human genome. So The optimist in me says, well, business is good. There's going to be a lot of loci that we need to do variant gene mapping on. But if every variant in every GWAS actually has multiple coordination of multiple genes, which is probably not the case, it's probably a subset, it means that there's a lot going on. And I think it's becoming quite clear to the field that we're going to have to do much more sort of pathway-based analyses, understanding individual susceptibility, understanding it from a polygenic risk score point of view, and really understanding the landscape and key intervention points in these pathways. And perhaps these sort of data sets where we're looking at genes and pathways, we can find those nodes for best intervention. So with that, I would like to just acknowledge my team and my colleagues at the Center for Applied Genomics, where we did so much of the original GWASs for the pediatric traits. And of course, I'm working with a series of collaborators on a different set of different sets of diseases, and particularly Gerd Blobel, who's a real epigeneticist and first pointed me towards TADS here at CHOP. With that, I'm happy to take questions. Thank you.
Thank you so much, Struan. That was great. If people would like to ask questions, please either put something in the chat or raise your hand. I can see it and unmute you if you just want to speak. Okay, we have a Yingji. I'm going to just allow you to talk if that's okay.
Yes, thank you. So such a wonderful talk. I really enjoyed so much. I have one quick question. In one of your slides, you said for the SNPs around the sentinel SNPs, you try to use very deliberately to include as many SNPs as possible for downstream for the variance identification. I'm wondering if you, what is your criteria? For example, you use a p-value cutoff at 5 at 10 minus 6 or something like that. Thank you.
Yeah, no, that's a good question, and we've tussled with that a lot. You know, when is a proxy? And you know, we've really dialed up and down on R squared, because obviously that's the main indicator of LD. And you know, we've played as low as like 0.2 and 0.6, but we're doing 0.8 now. I think that's the sort of generally recognized cutoff. And of course, many more GWASs are now reporting credible sets. And so if there's a credible set available, we'll give priority or we'll give extra weight to the SNPs in that proxy set that occur in the credible set.
Got it. Thank you. Thank you so much.
Thanks for the question.
Other questions? Eva has a question. I'll let you talk.
I think you can hear me. I guess, is there much more to be gathered by comparing what small amount of information we have on little isolated populations, maybe Africa, Brazil, in comparison to the large databases we have of European ancestry people?
Yeah, no, it's a good question. Of course, we know there's a particular skew towards European ancestry with GWAS findings currently. The positive about our approach is we've created this resource. We've done it on 54 cell types now, and we're ethnicity non-specific. So whatever variant is reported in whatever ethnicity, we can then look at the coordinate of that variant. We're not actually, our data doesn't depend on variants. It just depends on the location of where the variant is. And we ask if it's in an open region. So as I'm optimistic as more and more GWAS is done in different ethnicities come out, we'll be able immediately to be able to query that data and make inferences on the causal gene.
So I stumbled across something that had been overlooked by medicine, And it has to do with actually a hormone called aldosterone and blood pressure. And so it appears like far down in South America, there were people isolated inland. And so they were eating a lot of fruits and vegetables to survive and animal protein. And so they didn't have enough salt. And so I've discovered also the same variant may be present in Africa. And I will send the information to Maria Costanzo because we exchange emails and maybe she can forward it on if that's okay.
Happy to query our data sets to see if there's some lead there. So it sounds like, Eva, you're not sure what the causal gene is then.
We're not at the gene level yet. I'm hooked up with a group of endocrinologists online on Wednesdays, and I've nagged one of the endocrine physicians to help me a little bit. So I'll send it over to Maria and then she can forward it on to whomever. Is that okay?
That's great.
Let's see, Yingji, I see your hand is still up, but do you have another question or is it just still raised?
I think it's a legacy hand.
I think a legacy hand. Okay. Yeah. Okay. Any other questions? All right.
Mm-hmm.
Thank you very much. It was awesome.
Oh, I like awesome. That's great. Thank you. I'm glad it had an impact.
Yes, thanks so much, Struan. That was great. And yes, we have been recording this and we will make the recording available to everyone so people who missed it can see it and people can rewatch it if they want to.
Great, and very open to collaboration if anybody wants to reach out.
Great. All right. We really appreciate your time, Struan.
Thank you so much.
Bye-bye.
Bye-bye.